Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in science and technology, focused on creating a better tomorrow. Today, we have the honor of being joined by Dr. Kartik Sunagar, who is assistant professor and group leader of the Evolutionary Venomics Laboratory, located at the Center for Ecological Sciences, Indian Institute of Science, where his team investigates venomous animals as well as their venoms as a model systems to address a variety of basic but also broad questions in both evolutionary biology and genetics. Dr. Sunagar is an evolutionary geneticist, and a major focus of his research to date has been on innovating and really studying the potential for pan snakebite therapies through the development, discovery of broadly neutralizing antibodies in this area. Dr. Sunagar did his Ph.D. University of Porto in Portugal, followed up by postdoc work at the Hebrew University of Jerusalem in Israel, and his work provided really fascinating insights, not just into venom ecology and evolution, but also the dynamics of venom evolution across time, the evolutionary origin and development of various venom-producing cells, the coevolutionary arms races involving venoms, as well as molecular mechanisms, genetic bases of venoms, and ultimately the role of environment and ecological factor that drove the evolutionary of these different venoms. A lot of really interesting themes that we have not gotten to in the past, but a really exciting show. Dr. Kartik Sunagar, thank you so much for taking the time to come on the show today.
My pleasure. Thanks so much for the kind introduction, and it's absolutely my pleasure.
It's great having you, and I would like to start off, as we typically do, handing you the floor for a little bit, just to talk a little bit more about yourself and your background. If you could sort of take us back to the early days of how you got interested, not just interested in genetics and biology, but obviously this very unique area of venom. I mean, your PhD title was molecular evolution of animal venom, so it was a very sort of early theme in this. But I just happened, also to notice that prior to working with venoms, you were also publishing on leopard genetics, which I guess an equally scary set of organisms to deal with. But talk about the early days, if you would, for a little bit.
Yeah, sure. So I have always been interested in wildlife. So I thought I would, grow up and research wildlife and, pursue research, especially into the genetic aspects of these animals. But I didn't know what exactly. I thought about looking at population genetics of, these endangered birds that you find on Andaman Islands, to looking at these rare mammals in the Western Ghats. So this is one of the biodiversity hotspots in India, which is in the eastern part of India, stretches all the way from Goa to the southern part of India, Kerala. So it's a really long stretch of forest, and houses a lot of diversity of animals. So I was very interested in looking at some of the very rarer animals that you find only in that pocket of the Indian subcontinent. But later on, my interest in snakes grew, and I started catching snakes and looking at them and reading about them. And so what fascinated me a lot early on is, so basically all these venom components that you see in snakes and other animals, you and I also produce them in our body, but they are, they serve completely different purposes, and they're completely non-toxic. So it always fascinated me to learn or to understand how something completely inert becomes such potent toxins when produced by these animals. So I wanted to understand, what drew that transition and what are the molecular basis of that transition. And that's why I decided that I'll look at, the molecular evolution of venom. And as you correctly pointed out, that ended up as being the title of my thesis. So I was very fortunate to understand how venoms evolve in various lineages across the world. Um, but one of the other things that was relatively understudied, it's still understudied, is how these venoms are produced and how they are regulated. So I went on to do a postdoc at Hebrew University, as you suggested, where I was a Marie Curie fellow and a guest scientist. And we made these transgenic lines of animals, venomous animals called sea anemone, which are related to jellyfish. So we made them to produce, we genetically engineered their genomes so that they produce fluorescent proteins in cells that produce venom. So we could look at these cells, right from the earliest stage of their development, how they evolve or how they develop across space and time. So that was really cool, and it told us a lot of things. But then I had to come back to India because, in one of the things that probably will come out of this conversation today is that India suffers from the brunt of snake bites. Snake bite kills more people here than anywhere else in the world. So I always wanted to come back and, figure out better solutions for this problem. And so I started the Evolutionary Venomics lab at the end of 2017. And it's been 6.5 years, and now I'm serving this role as an associate professor here. And I have a really broad team of with a lot of students at various stages of their career, and we are all looking at very interesting questions, as you pointed out, especially concerning venomous animals and better therapy or designing better therapies for treating bites from these venomous animals.
Mm-hmm. Mm-hmm. And I thought it was, extremely interesting, because we talked about, for instance, plants on previous episodes, which are, also pretty good at creating interesting, complex mixtures of bioactive substances that, primarily used for defense. As you point out, these venoms are not just extremely complex cocktails of, in many cases, larger molecules, in terms of the proteins and so forth. But a lot of diversity, and again, going back to your PhD and the things that they do, whether we're talking about being cardiotoxic or neurotoxic or hematotoxic, and then at the same time, there's this really fascinating fact that you talked about, where it said that venom has sort of independently originated something like 100 times throughout evolution in different ways. And I'd love for you to talk about those two things, because I think it's very important to understand sort of, the complex nature of these materials via the unique evolutionary processes that are happening, and the fact that they originated many times along the last couple billion years. Talk a little bit about it, if you would.
Absolutely. I think, that's one of the most fascinating things about venom evolution. So as I pointed out earlier, we also produce these proteins. So we do have the templates for producing those toxins. So, because these are proteins that were probably recruited early on during the evolution of all kinds of animals, right? So most animals have these templates in them, where these proteins serve very many different functions, as you pointed out. They might be involved in immune system, they might be involved in reproduction, or just maintenance of homeostasis, right? So venomous animals at various time points have used those templates or those proteins and modified them further, especially by changing the surface residues. So this allows them to now target newer or interact with novel receptors, right? So that's how they have become toxic in the salivas of many of these venomous animals. Yeah. So it's the same template that has been used by all of these different animals. In fact, this is very much prominent in spiders. So one of our recent research projects basically shed light on how one of the major toxins in spiders, all around the world has evolved. So it turns out that all of them have evolved by modifying a single molecular template or a very similar molecular template, which was tinkered along different lineages into different structural and functional forms. just like you see in snakes and other animals, but it's just that they all started from one template. Whereas if you look at venoms of snakes or most other venomous animals, they have all used different proteins into the venoms.
Okay.
So there are many gene families that are included in the venom, but whereas these spiders seem to have just used one template or very similar set of templates to generate all the diversity that you see in them. So I think this is why you have such recurrent evolution of venom and even venom resistance in various animals, because all of them have those templates, and when they have to deploy it to support their ecology, right? Either it's feeding ecology or defense. They've all seemed to, tinkered with the same set of proteins that are expressed in their salivary glands or other tissues.
And you are right now. I'm sitting here on the east coast of the United States. Not a lot of snakes. Well, maybe some, but some local ones, but nothing extremely venomous. But you are in India, and you have written about the big four that you contend with in your area of the world, namely the speckled cobra, the common krait, the Russell viper, and something called the saw-scaled viper. And these are responsible in India alone for something like 46,000 deaths annually, about 140,000 disabilities based on snake bites. We'll get into a little bit about sort of the early antivenom development history and so forth, but talk just a little bit about sort of I was called the issues with these and other venomous snakes in India, and sort of the broader situation if we look at global WHO data with regard to what we're dealing with just with snakes. And I guess you can throw in other creatures there, whether it's scorpions or spiders or jellyfish, what have you. But what's the current 2024 situation with regards to death and disability of all sort of venomous creatures that we're looking at?
Yeah. So I think, you basically, mentioned it. So it's not 46,000, it's actually 58,000 plus now. I mean, according to the recent estimates. And the 140,000 statistics comes from the fact that it's estimated that the number of morbidities is usually thrice as high as mortality.
Got it.
But again, we don't know the exact numbers. We don't even know the exact numbers of bites or deaths. I mean, if I just talk about India, we don't have accurate numbers for that. These are all estimates based on studies that have taken place in the field, where they have contacted people and use statistics to estimate, basically extrapolate what would be the actual number of snake bites in the Indian subcontinent. This is mostly because snakebite is not a notifiable disease. So what happens is in case of, so the hospitals don't have to notify to the government that they have admitted a snakebite victim. So that's why those counts are not officially kept in most hospitals. So that's why we don't know the exact numbers. But in the state I am located in, in the state where Indian Institute of Science, where my lab is located in, called Karnataka, here very recently, in fact, last week, snakebite was made a notifiable disease. So, hopefully in a year's time, we will have an accurate number of bites at least from this part of India. But yeah, I mean, soon it's around the corner. I think most states will soon make snakebite a notifiable disease, and we'll have an exact number of bites from various species. So the problem, if I just speak about India, I mean, this is applicable to sub-Saharan Africa as well, is that, there are many more species than just these four species that you mentioned, the spectacled cobra, Russell's, saw-scaled viper, or the krait. These are supposed to be responsible for most of the deaths and disabilities, but that may not be true. So Russell's viper is definitely the one that kills a lot of people, but it is actually followed by krait, whereas people think it's spectacled cobra that causes a lot of bites and deaths and disabilities. But it turns out that it's probably krait that does that. But if you go to some parts of India, like if you go to the northeast, right, you will not find many of these big four representatives. They are actually replaced by other species which are closely related to the big four. Like you will find the monocellate cobra, you will find various species of kraits there, except the common krait. I mean, in most parts at least, then you don't find Russell's vipers in most of the regions in Northeast India. Then there are various species of pit vipers which are found in those dense forests, and mountain pit vipers and so on, which cause a lot of morbidities and probably even deaths. We don't have the statistics so far. So this is very true if you know which part of India you travel to. There are many species that are only regionally prevalent, so they have this restricted geographic distribution, and that's why people don't really worry about them. They think that this antivenom will confer protection against even those species because they're many of them are closely related to the big four. But through our research over the last 5-6 years, we have very clearly seen that the antivenom doesn't actually solve this problem. And again,
Sure.
we can get into that in a bit. The technology is very old, so the current antivenoms don't really cater for that, and that basically compounds this problem further. Um, and the other problems include, people not reaching the hospitals on time,
Right.
because many of them are located far away, and even the landscape doesn't permit them to reach faster. So even if India becomes a first world country in the next 10 years, some of these places are so remote that either you destroy those forests, or it's just very difficult to get those people to reach the nearest hospital very sooner. It's just that India has to expand our medical facilities significantly. Only then I think some positive change would come in future.
Mm-hmm. No, you just mentioned about sort of the quite antiquated nature of antivenom development and manufacturing, in the sense that the current method is still based on this equine. So we put the venom in a horse, and it makes a bunch of these polyvalent sort of non-specific antibodies, and that's sort of, as you point out, it's sort of been that way for about 100 years now. You're going to be changing things significantly, but let's just talk a little bit about that current situation that we have that's been, as you say, unchanged for about a century, and what current antivenom products are sort of based on, and a lack of, again, specificity, which, as you point out, there's very specific differences between, the venoms produced by these different snakes, and that can make a whole lot of difference, again. And we'll be getting into your recent paper and where you're going with all this, but talk about the past here as well, just to get people on the same page, understanding where we're going.
Yeah. So as you mentioned, the technology has not changed in over 100 years now. We still use the same methodology of delivering sublethal, and it's not subtoxic, but we would like to believe that it's subtoxic as well. Subtoxic doses of venoms into horses, ponies, and mules. Those are mostly used. So it's an equine product, as you correctly mentioned. So these are administered into these animals. Those animals would recognize these antibodies as foreign antigens and produce an immune response against them. And so these antibodies, resultant antibodies, are collected, they're purified, and that's what is sold as the treatment of snake bites. But these animals face a lot of pathogens during their lifetime, just like us, and they would also produce antigens against those pathogens. There are lots of toxins which are not very specific to humans, because snakes produce these toxins to target their natural prey or predators, right? Humans have nothing to do with them. So we don't need all of those antibodies. We probably only need like 1% of or 10% of those antibodies that are produced. Right. In fact, it turns out that only 10% are specific to snake venom toxins. The rest of it is just therapeutically redundant to humans. So what this results in is, you require many vials of antivenom for treating snakebite victims, because the dose effectiveness is so much lower, right? Because very smaller proportion of these antivenom is actually targeting the snake venom toxin. So that increases the cost. In some places around the world, the cost of a single vial of antivenom can, exceed $100, where these people earn less than $5 a day, right? So it's just not affordable, and they will obviously require many vials for treatment. It's not just one vial that's going to suffice. So it not only increases the cost, but it also increases the risk of these people suffering from secondary reactions to the antivenom, including anaphylaxis, which can be fatal. So the antivenom itself can kill if you inject very large amounts, considering that it's an equine product. So yeah, I mean, the changes in antivenoms are urgently needed. And as you also kind of mentioned, that it lacks specificity, it lacks that broad effectiveness. So what we were discussing a couple of minutes ago, that there are many of these other medically important snakes for which there is no effective treatment, right? So like the king cobra, we don't have an antivenom, although bites from king cobra is very rare, but still, zookeepers, animal handlers, or people who like to keep king cobras as pets, especially in the Western country, they don't have a specific antivenom against king cobra, especially if this king cobra comes from the Western Ghats of India. We have one of the most toxic snakes in the world called syndicate, which we have now discovered even in some of the southern parts of India. So, and we don't have an antivenom for that. So you definitely need a very an advanced therapy which also offers a broad spectrum of neutralization and works against a lot of these different species. And it's not just the species that we need to worry about, it's also the populations of these medically important species. So the first two toxins that you mentioned earlier, the neurotoxins and cardiotoxins.
Right.
So if you look at venoms of cobras in northern India, a lot of them have, especially in the western part of India, a lot of them have venoms rich in cardiotoxins, which are also called cytotoxins. So what they do is they cause a lot of necrosis. They result in a lot of morbidity. So if you go to these places, a lot of people that we have spoken to suffer from, amputations and disfigurements, which not only puts them out of work, but there's also a lot of psychological burden, which is very neglected. Nobody has studied the psychological burdens of snake bites, especially in this part of the world. So, but if you come to the southern part of India or the rest of India, you'll actually find the same species of cobras, the spectacled cobras. They start producing venom that is rich in neurotoxins, which targets a completely different system. So yeah, you need a product that not only takes care of venoms of different species that are medically relevant to humans, but also the different venoms that the same species could produce in different parts of India.
Okay, so that brings us now into your laboratory. And I was wondering if you could, before we get to the broadly neutralizing work that you're doing, just talk a little bit, introduce us to your lab. So your lab is, as I mentioned, beginning the Evolutionary Venomics lab in the Center for Ecological Sciences, part of the Indian Institute of Science, which but your funding, it not only comes from local institutes. You have partnerships now with Wellcome, the Wellcome Trust, the UK Department for International Development. So a lot of attention giving from both India and abroad on what you're doing. Talk a little bit just about your lab there, your department, what type of staff you have that you're overseeing, and then I want to get into the project that was just in the news.
Sure. So we have so there are three major areas of research that my lab focuses on. And we have students from all stages of careers, all the way from interns to masters and bachelor students and PhDs and postdocs and senior scientists. So I have a pretty large lab. We have roughly around 40 people in the lab at any given time. But that gives us a lot of opportunity to look at many different questions at the same time. So a lot of us, I'm primarily interested in understanding the evolution of venom. So I'm very driven by projects or looking at questions that are evolution oriented. So understanding, how resistance to venom evolves in prey, how resistance to evolve in predators of venomous animals, and questions. Most of the questions that I have looked at in the past. Then the other half of the lab mostly focuses on snakebite because of the reasons that I mentioned. That there are very few people in India looking at developing solutions for snake bites, unfortunately, although it's our problem. So half of my lab is, specifically looking at snake bite as a problem and trying to figure out, many different ways of addressing this problem. So one of the things that we'll probably discuss is the antibodies, right? So these synthetic antibodies that we can make, but they're also looking at existing drugs in the market, which have been useful in treating cancers and other types of diseases where the targets might be very similar to what you find in snake venoms.
Yep. Yep.
And then there is also a small group that's looking at these medicinal plants. So India is also known for Ayurveda, right? The use of these
Sure.
traditional medicine. So a lot of tribes, fortunately or unfortunately, rely on these medicinal plants without even knowing, how useful they are. So we are also looking at determining the usefulness of these plant extracts and trying to figure out if there are any compounds in these plant extracts that might be useful. And we have already found a lot of interesting results there as well. So yeah, I would say the lab is interested in understanding the ecology, the evolution of venoms and venomous animals, looking at their, even evolutionary histories. A lot of my PhD students are trying to understand how did these venomous animals evolve or diversify in India, right? I mean, for example, if we talk about spectacled cobra, you find them all the way from Himalayas to, the southern tip of India, right?
Right.
But we don't know where did they originate. Did they originate in Africa? Did they? I mean, there's a lot of indications that that's what happened. But
Mhm.
even to understand, how did they enter India? How did they diversify? How many lineages got into India? Just like how people have been trying to understand how humans have evolved, right? How they have migrated, how many
Sure.
lineages have migrated in and out of Europe and Asia and Africa and so on. So you can ask all of those very interesting questions with respect to these animals as well, because understanding their evolutionary history is also very fascinating. Because it's not just, I mean, they're not very boring like humans. I mean, they also have these very different venoms, a lot of very interesting compounds in the venom. So yeah, a lot of my students are also looking into those aspects. So I would say it's a blend of ecology, evolution, and snake bite therapy.
Excellent. So with that, actually, let's start with the very recent paper that was in the news, synthetic development of a broadly neutralizing antibody against snake venom long chain alpha neurotoxins, just published. And again, in this research and through this paper, you're basically advancing the science of the whole area about 100 years at one time. But again, developing synthetic material, looking, at libraries, humanized antibody libraries to neutralize this very important part of the neurotoxin. I understand. Take us into this work, because I think it's extremely, an extremely core output to everything that you've been doing the last many years.
Absolutely, I would love to, but I would like to give you, background on how all of this started first.
Sure.
So basically, as you pointed out earlier, a lot of these projects are funded by Wellcome Trust, where we have been immunizing or basically harnessing, unique immune systems of various animals, like camels. Camels produce these very single chain antibodies. Right. So instead of two chains, they produce a single chain.
Yep.
And the advantage is that, it's just a small part of that single chain that is of therapeutic relevance, meaning that's the one that binds to the toxins or any antigen of your interest, right? So you can produce them as nanobodies. So they are called nanobodies, where you just produce the smaller part of that fragment. So if you can think about it, it can actually help in treating morbidities, because it is a small molecule, it can penetrate tissues better and probably would serve a greater role in countering snake bite related morbidities. So one of the major projects involves immunization of camels with venom toxins that cause morbidity or that cause necrosis. And then what we do is we have been collecting blood from these camels. So we have been immunizing camels in Rajasthan, which is in the western part of India, the arid region. But we have collaborators as part of this big project of the consortium that we call Serpents, which involves my lab at IISC, Liverpool School of Tropical Medicine in the UK, Scripps Research Institute in the US, and International AIDS Vaccine Initiative in the US, and then a couple of partners in Africa. So that's how it all started, where we were immunizing camels here in India, immunizing camels in Kenya with our good friend George, immunizing baboons, cows, because all of them have very unique immune systems. So the idea was that, you could harness or harvest their RBC, their blood samples, and then isolate memory B cells from these blood samples. and then basically use synthetic toxins as baits, to fish out. So basically, antibodies are produced by these cells called memory B cells.
Mm-hmm.
So what they do is they display these antibodies on their surface first before they secrete it out into the plasma, where it encounters the viruses and bacteria and neutralizes them. So what we can do is we can basically use toxins as baits to fish out those memory B cells that have antibodies on their surface, which is very specific to the antigen of our interest. So
Mm-hmm.
this antigen can be a toxin, if it can be HIV, it can be COVID. So it depends on what do you want to find antibodies against, right? So whatever that antigen may be, you use that, you fish out these cells.
Yep.
And once you fish out those cells, you can sequence them. And once you know the sequence, you can basically express them in mammalian cell line. So you can produce that antibody in mammalian cell line in the lab rather than relying on an animal. So in future, you don't have to immunize those animals. You just keep producing these cell lines, which will continue to produce these antibodies for you. And once you get those antibodies and you purify them, you can now test if those antibodies are, they also have neutralizing capabilities, right? So this is a major, aspect that's happening in the lab, and we have been discovering a lot of interesting antibodies. But this work that you spoke of is basically a collaboration with the Scripps Institute, and a lot of credit goes to Joseph Jardine, who's an immunologist and an expert antibody engineer. So what Joe managed to do, so the other approach to discovering antibodies is using these human libraries. So
Right.
basically, when we are producing, I told you that, these memory B cells produce these antibodies on the surface, right? But what we could also do in parallel is we could also take those blood samples from these animals that are immunized and then convert them into, a library of yeast cells.
Right.
So this yeast cell library will basically display these fragments on the surface, the antibodies on the surface. And then you repeat the process that I said, where you use antigen of your interest and pull out yeast cells that are coding antibodies specific to the antigen, either venom, HIV, COVID, whatever it may be, right? And then you plate those yeast cells and you can continue to produce them in large amounts and forever, right? So you don't have to go back to those, you go back to animal immunization ever again, right? So you can keep discovering these antibodies. So basically what Joe did is he used a library of those yeast cells that were producing human antibodies.
Mm-hmm.
And so we basically designed antigens that might be of interest which we could target to knock out the toxicities of this large group of medically important snakes called elapids or elapidae. So it includes snakes like cobras, kraits, king cobras, sea snakes, so on and so forth, brown snakes in Australia, so on and so forth. So so what we did is we used many different, so one of the major toxins that these elapid snakes produce is called a three-finger toxin. It's called so because it looks like three stretched out fingers. So we used many different versions of those three-finger toxins and basically pulled out these cells that were coding for antibodies that are very specific to that toxin. And then, so that's how we kept refining them. And you can also introduce mutations in these antibodies to make them much more efficient to the target. And so that's basically resulted in identifying a bunch of antibodies that were very specific to these three-finger toxins. But one of them was very broadly effective. It could recognize three-finger toxins from elapid snakes around the world, like the king cobras in the Western Ghats, the monocellate cobra in Eastern India, the multi-cinctus krait in Southeast Asia, or many-banded krait in Southeast Asia, and then the black mamba in Africa. So that was really promising because you now have an antibody that basically a single antibody that protects mice from complete effect of envenomation, and that too from snakes that are found on different continents, right? So all the venom variation that I spoke of earlier, I would have imagined that it would be impossible to design something that would work against different populations of the same snake, maybe, but not the snakes in different continents. So this shows that you could eventually get a universal antivenom therapy, which would work in any part of the world and would hopefully work against most of the medically important snakes. Sorry for the long answer.
No, it's an awesome answer, and again, because we've been talking about this pan theme in the past, especially when it comes to pan coronavirus vaccines or other sort of zoonotic stuff. But in this case, I mean, the story you're telling, again, and not as much not just advances the science a century, but it just it provides a solution to something that as you pointed out as we walk through the history here has been significantly lacking. So no, that's extremely exciting. One thing I wanted to ask you, along the lines as you're looking for these pan antivenoms. There was an interesting paper a few years ago in the literature that you put out. It was entitled "Scratching the Surface of an Itch: Molecular Evolution," in this case of aculata venom, which I guess comes from certain species of ants and bees and so forth. And again, here you were talking about, I guess, within the venoms, obviously there's the really nasty toxic stuff, but then there's the other stuff that causes all sorts of other problems in terms of the allergies and the hyperimmune response, and that's a whole other area of interesting sort of pharmacology, let's say. Anything going on of interest related to that? Because that's while some of that stuff might not kill us as much as the other as the poisons, still there are as many important targets there for other sort of unmet areas of anything interesting going on there in terms of sort of the pan strategy and sopping up some of those moieties as well.
So some of these technologies that we have set up in the lab can be used to answer those questions or identify solutions to those problems as well. It's not just limited to that. Even we frequently get outbreaks of Nipah virus,
Yeah,
very close to where I'm located now. Coronavirus obviously has been a big problem, for everyone around the world. So you could use this technology to discover antibodies against any antigen or target of your or antigen of your interest.
gotcha.
But I mean, there are lots of things happening in terms of bees, wasps, and ants in my lab, but most of them have to do with the evolution of the venoms that they
Got it.
produce. To understand. So, for example, if you think about bees, right? I mean, bees have so many different castes. They have queens, they have nurses, they have toddlers, where you
Yeah,
just want bees, and they have soldiers and foragers which go out, right? So we are trying to understand how venoms in all of them evolve, because only some of them are exposed to predators, whereas others are most of the time inside the nest and don't really get exposed to predators, right? So we are trying to address questions like that mostly, but not really looking at it from a perspective of them causing allergies. And but you're absolutely right that they not only cause minor allergies, but they can actually be fatal. A lot of
right.
things that happen. Again, because we work with clinicians around India who treat snake bites, they also treat other emergencies, right? Medical emergencies, including bee stings and wasp stings.
Anaphylaxis.
And a lot of times, the bee sting patients that they get, they have hundreds of bees stinging them. The bees would get into their noses and mouth and ears. So it's a very horrible scene to look at. But yeah, I mean, they do kill a lot of people,
Yeah,
although it seems like it's just a minor reaction. And what's amazing is, so based on what we have been doing, it seems like they have some components which basically create an avalanche of, basically a cascade of reactions in the target. So if you think about snake venoms, we have extracted close to 500 milligrams of venoms from a single individual. If you look at African snakes, you can get more than a gram of venom from a single individual,
mm,
where only a microgram is enough to put you
right.
into emergency, right? So they produce so much of venoms. Whereas if you think about bees, they produce such tiny amounts. You get hardly like 2-3 microliters from a bee, from a
Right.
single bee, right? So they have evolved ways in which they can, even with such small amounts of venom, they can cause all of these, interesting and lethal reactions into the target. So it's amazing how they have done that.
What's let's go to the other side of this equation, because as you know, and again, you pointed this out in one of your publications, and we just were chatting about it on a recent show, that I came out of the pharmaceutical industry, and there's quite a few billion-dollar plus therapeutic classes like the ACE inhibitors, like the original IGF one diabetes drugs, even a lot of the thrombotic agents that had their origins or their skeletons, let's say, from some of these unique venoms that were discovered out in nature and then ultimately connected into sort of a interesting pharmacotherapeutic opportunity. Anything exciting popping up on your radar as you're studying these unique venoms and their evolution as far as that other side? Because clearly there's a tremendous amount, whether it's non-opioid pain relievers or whatever the case may be, that these species may be pretty damn good at creating biologically active substances for. What are you doing in that area?
Yeah, so one of the advantages of having the large lab, as I said, is we can look at all these very, very different questions at the same
Right.
time. So we have also been looking at animals that are very least studied. For example, things that we usually see around us. So there is a species of Porto wasp that we always see here. So we
Okay.
wanted to look at its venom, and we actually found something very interesting that basically acts as an extremely potent anticoagulant.
Yeah.
So it completely prevents the blood from clotting for more than 30 minutes, which is like the upper limit of our instrument.
Yeah.
So we haven't done much there. We are trying to figure out what is this particular component, how we can engineer it to harness this property. Because this anticoagulant effect, as you're in the pharmacological industry. So it's also very useful in certain scenarios, right? So that's something related to biodiscovery. The other thing was scorpions that we see again in a backyard. So we were looking at the venoms and trying to see if they are very specific to certain strains of insects, that are basically pests of economic importance, right? That basically infest crops of economic importance.
Sure.
And again, we found certain fractions which seem to contain toxins that only affects them. So now, so we are looking into harnessing that property to make these bioinsecticides, right? So one of our good friends from Australia, Professor Glenn King, so they basically recently made the first commercial spider venom derived bioinsecticide in the world, which is
Wow.
absolutely amazing, right? And these are small peptides, so they're very easy to synthesize. They are very stable, so you can spray them. So we have done that as well. So even if you spray them, they last for a very long time because they have, unlike snake venoms, they have small peptides and
Sure.
they are linear peptides. So these caterpillars could feed on them later. And what we find interestingly is that many of them die, but some of them survive and pupate, but they never come out of that cocoon. They just die at that stage. Or if they come out of the cocoon and pupate, then they have all these deformities. So ultimately, the venom is affecting their population. Either by killing them or making them so deformed that they would be easy prey for predatory birds and other animals. So I think there's so much to look at even from that perspective. And unfortunately, again, I think very few labs. I can't think of any lab in India that's looking at all these amazing venomous animals that we have in our backyard.
Yeah,
So even
absolutely.
cancerous cells, right? I mean, many components can
Yep.
specifically target some cancerous cells.
Yep.
We don't know how they do that. They probably target a receptor that's overexpressed in cancerous cells compared to normal cells. But yeah, that's another fascinating area which we haven't gotten into yet because as if we don't have enough questions to worry about at this stage.
Right.
But you are absolutely right. That's an amazing field of its own. Biodiscovery, discovering all these compounds of therapeutic or economic importance from these venomous animals. And I think we are located in a great place. As I said, these animals have never been looked at from any of these angles. So I think we're all sitting on a gold mine, and we keep looking. Hopefully, we'll find something very interesting someday.
And one last thing while we're on it, because we talked a lot about the sort of the genetic evolution of these materials, but you also study the ecology, the environment, and I don't know if you spend much time with them, but I remember the talking a couple of years ago about these poison dart frogs, which they have to eat certain insects. that have eaten certain plants, and there was a whole chain of things that by itself the frog was pretty normal, but after it ate the sequence, it became the most poisonous thing that was out there. Any interesting fun facts that we should take away, whether it's the frogs or any of the other unique creatures that you're dealing with, that we can go back and maybe tell our kids tonight about?
I mean, a very interesting and very popular parallel example to that is, the sushi, right? The fugu that,
Oh yeah. Yep.
Japanese people eat. That also gets its poison from, what it eats, right? So if you raise them in the labs or in aquariums where they don't eat the natural prey, then they are also completely non-toxic. We have something similar here. I mean, these are very rare animals, right? That fall into this category, where they derive these poisons from what they eat, right? We are actually looking at something very interesting. It's called, these are buffonid toads. So these toads
Yep.
produce cardiac glycosides, which
Yep.
basically, they bind to your heart muscles and cause cardiac arrest, right, heart attack. And
Yep.
they're extremely potent. We have had unfortunate incidents in the lab as well, but thankfully that was not potent. That was not significant. It was just a minor accident. Um, but yeah, these toad toxins are extremely dangerous, and so we are trying to look at how various animals have evolved resistance to this toad toxin. So there's something very interesting that has come out there. But yeah, there are some snakes which are not only resistant to toads, these toad toxins, so they can feed on these toads. But as just like the poison dart frogs, they also store those poisons in their nuchal glands. So these are glands at the back of the snake, snake's neck. So when you touch them, they release this poison. So this poison can be absorbed by your skin, and it can put you into cardiac arrest. The snake, to make matters worse, the snake is also extremely dangerous. If it bites you, it has very potent venom that can definitely put you in a hospital. So this is an example of probably one of the very few examples of animals that are both poisonous and venomous. So the distinction being that venomous animals are capable of, um, basically inflicting toxicity by, through a mechanical way, right? Either by biting or stinging or some other way of injection. Whereas poisons are passively absorbed, right? Like poisonous gas or a liquid that you eat, or the poisonous compound that you eat, right? So that's the distinction. But this species can bite you, so it's and put you in the hospital, so it's venomous. And if you touch it, it can release that poison, so it's poisonous. So one of the very few examples where an animal is both poisonous and venomous.
Very cool.
But besides that, we don't really deal with anything that's mostly poisonous.
Got it. Very cool. Kartik, while we have you, I noticed a couple weeks ago there was something known as SnakeSemp 2024, focused on which is venom pharmacology and drug discovery, that you attended. What else is coming up for you in 2024 that we should know about, per conferences, talks you're going to be giving, any other initiatives that public facing while we have you that you can mention right now? Anything else you want to mention while we have you today? It's been very exciting, but please.
In terms of conferences, I think there's the World Congress by International Society for Toxinology, which is a world event, and all toxinologists get together. So this time, it is being hosted by Professor Manjunath Kini, one of our senior veterans who's been working on venoms from a very, very long time, even before I was born. So he's hosting that in Singapore, and all the toxinologists from around the world would attend this meeting, and I think it's going to be a very exciting meeting. So that's definitely going. I don't know if they would make it open to public because it's a registration only, but that's definitely a big event where all these exciting talks would be presented. So I'm also presenting our work there. So I'm really excited about that. But besides that, there's this Cordon conference that's happening in your backyard in Mainz, I think.
Nice.
That's happening in August. But yeah, again, I don't think that's public. I think that's only for a subset of toxinologists to attend. I think it's invitation based only. But yeah, that's another thing. But yeah, I haven't really come across any other conferences this year.
Got it. Excellent. Well, I mean, it's been an exciting few years. Again, I've enjoyed watching the recent story in the news, and we'll continue to follow everything you're up to. It's a really exciting space, both in terms of the countermeasures, but also the potential for novel drug discovery. Again, for everybody that's going to be listening to this particular episode of our show across the various podcast networks or watching on the YouTube channel. Again, you've been listening and spending time to Dr. Kartik Sunagar, assistant professor and group leader, Evolutionary Venomics Laboratory, Center for Ecological Sciences at the Indian Institute of Science. Kartik, I want to again thank you for taking the time out of your schedule. I know it's late in the evening there, but thank you for taking the time to educate us on a little bit of what you're up to. Obviously, thank you for what you do. And as we like to say on our show, thanks for helping to create a better tomorrow via the type of science you're working on. It's a really great story.
Thank you. Thank you so much. You have been very kind.